10, and we're going to continue to wrangle our data, the microbial data from the Tara Oceans project. That's right where we left off last class. So recall now it's been 2 days, and Les Stroud, our survivor man, has still not caught any fish or microbes or water, and it's becoming critical that we finish this today. We're still right here in the tidy universe, in the tidying procedure, and getting this data set all ready for our analysis. So today we're going to do that, and then we're going to do a little bit of visualization and exploration of the data. Okay, so we're going to cover two functions. One's called separate, the other's unite. They're contrapositives of each other. And then some set theoretic notation, inner and outer joins, a little bit of normalizing the Tara Oceans data set, and then visualization using heat maps. Okay, so let's go back now to our studio. I should warn you that this lecture is highly technical. It's really in the grime of wrangling. I hope that I would recommend that you keep your RStudio session open and pause the video and follow along step by step with me to make sure that you understand the individual concepts. Okay, so let's leave it there, and I'll see you in a second. All right, this is where we left off last class, and I'm going to continue in the style of just walking through what I would do. Okay, so here we had just put in this information in this little R chunk, and we were looking at the class of different fields returned by the parser. This was that heuristic function, and so it guesses the station identifier correctly as a character, recall, and it guesses that this field called date time is actually a date time, and that's R has a class for holding both the date and the time. Now, in some contexts, we might want to separate the date from the time in one value right now. They're pushed together. This is a bit of an artificial example for this data set. We aren't going to actually look at the date and time separately, but sometimes you do have this kind of situation where the data has been kind of glued together, and there's a way in the tidyverse to separate that kind of information, and not surprisingly, it's called the separate function. So first I'm going to go back to here and just orient you. If we go back to our tidy rules, one of them was that each value must have its own cell, right? So in this case here, it's a violation of the tidy principles because both the date and the time are in the same cell. So we want to separate those out. Okay, so the separate function, to come back over here, would take something like this, and I printed it out here, date and time. Now, if I want to separate them, I would pipe my table into separate, so implicitly T1 is here. And then there's two other parameters. Well, okay, firstly, this is the column that we want to actually split or separate, and then two other parameters. The firstly is into, and now what this means is that I'm giving it the names of the two new columns it's going to create, date and time. And the separator here is going to be the space between them. So when I run this and I pipe T1 into this function, my table now is going to have a date column and a time column, and it's going to have the date part in this one and the time part in the other one, as you would expect. So we can do that, and I can run it over here to show you. And when I do that, whoops, oh, sorry, I had to, okay, right. So I'm going to read that in. I forgot to execute that function. So now when I pipe T1 into there and I save it back, I get some warning messages. Let's forget about those. They're not important. So here, I want to make sure it's clear, is I'm piping T1 into the separate, this function here, and then the result of all of this expression from here to here is assigned back equal to T1. So in a way, I'm updating T1, and now when I look at T1, I can use the view function and come up here. Now if I scroll along, I will see in fact that there's now a date and a time, and that old date time column has disappeared. Okay, so that's kind of nice, but there's a problem. And the problem here is that if I look at T1 quickly, and I come down here and I find date and I find time, you might astutely recognize that now are things that they're characters and not actually date and time. And there's actually a class called date and a class called time. That's not optimal. So the separate function lost actually the class of the data and defaulted to a more general class of character. Everything in R seems to default to characters when it can't make sense of them. So we have to fix that. This is a classic example of data wrangling, right? It's so boring sometimes, but it's so important to get the data tidy, right? To get it all set up. So here, if I run the guest parser, this heuristic on those two fields, in fact, it gets them right. And if I parse the date, it's going to parse it into, in fact, date. So I could ask, like here, I could ask the class. I can ask the class what's returned by that, and you'll see it actually does find them to be dates. So what I can do is I can execute this kind of expression. Here, I'm going to update T1 date, right? So that's the column date that already exists by parsing, parsing the parse date of T1 date. So basically, I'm just going to update T1 date, and now it will actually have the right class. And the same thing for time. I just basically assign T1 at time back, and now when I look at T1 and I scroll down here to date, it's actually now its class is date, and time, its class is time. So it's a small inconvenience. Okay, so some people might have questions as to what these symbols are, and that's a language called LaTeX. It's a way of embedding fancy scripting, scripts and fonts and boldface and mathematics into your text. It's not important. It's not part of the course. It's in our Markdown. You can put LaTeX in, and it'll compile when I hit knitr. So I can actually show you that right now before we go on. It should be under here import.html. It's the output of my session. Oh, it's a little bit big. I'm not that old yet. So you can see that this is what the R Markdown file looks like. And you can see we have our links in there, and we have our code interleaved with our natural language text. And maybe we can even, that's the output of the different R chunks. And where were we down here? We were even further along, a little bit further, I believe. It's quite long with all the output there. Here we had done the guest parser and the time and date. Yeah, and so, for example, I wanted this like kind of like computer text. So that's what that code means, was this, give it a different font in there. Okay. That's just for your knowledge, your interest sake. Okay, so there is actually a function that's the complement of separate called unite, and not surprisingly, unite. I don't know if I have a slide for it here. No, I don't have a slide for it. But the intuition, and there's no examples of it that I could find in our Terra Oceans data set. But basically, when two or more columns are separated, you can unite them into one column. Okay, so I don't have an example for that, but that should be okay. But generally speaking, the tibble now is looking pretty tidy. I can come in here and I can look at the column names, and well, some of these column names maybe are not the greatest, right? They could be cleaned up a little bit because they're quite long. And if you recall back to the breast cancer data set, some of these long labels really messed up the graphics, right, when we wanted to plot. So let's simplify these names. I mean, they for whatever reason, Tsunagawa et al. Needed these long names to be very clear what they meant, but for our analysis, it's nicer to have shorter ones. So let me give you an example here. This is one way to do it. I would keep around the old names. I'll call them old titles, right? And that's just to keep around what they originally were. And that's just the column names of T1, right? That's just all these guys, right? All these guys up here. But you know, all these like little ticks, etc., they're annoying. They're all these special symbols. You can never remember them. So now what I'm going to do is old titles. It's a vector of characters, and I can name that vector of characters. I can use the names function. So call names is for a two-dimensional matrix or tibble. You have columns and rows. Names is for a one-dimensional vector. Now here I'm going to just assign it like this. These are names that I made up, and I can show you what it looks like. Now if I look at old titles, well, now you can see that if I scroll back, the original name was sample label Tara station environmental feature size fraction, and I've just simplified this to be sample label. And before it was INSDC sample accession numbers, I simplified it to AC sample. Okay, so now what I can do is I can update my tibble T1 with the new names. So if I do that and I ask what T1 looks like, it's going to look a little bit better, right? Now it's sample label, AC sample, AC run. All these guys are on there. So it's a little bit improved. Now, I don't like, though, that I included here two things I don't like. Firstly, I have spaces in my thing, so it's sample space label. And that's sometimes a problem because, as you know, identifiers in R are single words without spaces. And so you always have to put these back ticks to say that it's like one character and not two. And so there's a lot of those. So I don't like that. And the other thing I don't personally like is using capital letters here, because you never remember whether at least me, I can never remember whether I capitalized it or not. So when you're programming, it's like just one extra thing to remember all the time. So what I've done here is I did exactly the same thing what I did above in the previous example, but I made better names. So here I'm going to use sample underscore label, AC underscore. I don't have any spaces anymore. And on top of that, I took away all of the capitalizations. And now when I look at T1, right, now you can see that all those backticks are gone away. I don't need them anymore. And they're short enough that you can see pretty clearly what each column is delimiting. Okay, so I find pretty good style. Now, you might say, well, but I should be using the full name in my figures, etc. And of course you can, because you can use the XLab or YLab labeling features in ggplot to overwrite now your made-up names. And because you still have the old guys around, old titles, you can always go back and get the original names. So I think that's pretty good style. Okay, so another little problem in our that's what I'm explaining in here is the other problem is that associated with these fields called size lower and size upper, and that's a bit annoying. And I just want to I want to show you what they look like. Size lower. This is the size of the filter they use to get rid of the small organisms in the sample, so the viruses, etc. Sometimes they used a 45 micrometer filter, sometimes 0.22, and then they have this less than sign. In fact, I find the best way to do this is just say ask for the unique guys. So this was what called the size lower, and there's 4 of those guys. Now, this less than sign, we would have to go back to the Sunagawa paper, but that less than sign, I guess it means that they used a filter less than 0.1. So it's not a number though. So these things aren't ordered properly either, and well, that's a bit of a problem because these aren't being treated as numbers now. With the double quotes, they're being treated as characters. And well, I guess that could be okay. But if I look at size upper, which is the size of the filter used to filter to get rid of the big organisms, you can see that they're actually numbers, right? And I can show you here, T1 $ size upper. I can see that there's something like 5 different sizes that they used, all the way up to 3. and down to also 0.22. Okay, so there's a bit of an inconsistency when the class of T1 upper is going to be numeric and the other one is a character. And so to make everything consistent, I would probably do two things here. First, I would make them into factors. And factors are something that's extremely important in R and the tidyverse, but they're important in everywhere in R. And we haven't talked about them nearly enough yet, so I want to talk about them a little bit now. And we're going to leave the Tara Oceans example. I'll give you something a bit simpler. So factors are this way of enforcing a dictionary on a variable. So it's a way of enforcing that a variable can only take certain values. That's not the same thing as the class of the variable. Let me give you a concrete example. We could say there's nucleotides A, C, G, and T, and I can put that into my session, okay? And I could think of a site, let's say a binding site. So this is where a transcription factor binds to a nucleic acid sequence and regulates expression of the downstream target, right? So sometimes that binding site for the transcription factor might be between 8 and 32 nucleotides long. And this is a toy example, but here's an example of a nucleic acid sequence that a specific transcription factor might bind, okay? Whoops. Okay. GA, small g, that's a typo. Should have been a big G. I missed the space, the shift. CCG, exclamation mark, I don't know what I was doing, and the T. So there's two spelling mistakes in there. Well, firstly, I could make this a factor. I could just say factor site, I'll make it a new variable, is set equal to parse as a factor, parse factor, site. So if I do that, the first thing, that's a good first step. Come down here. When I do that, I see that now I have two things here. I have the output like this, which is just the same thing as what site was equal to. But now I have this concept of levels. And levels are the values that variable can take. And by default, when I didn't specify a second parameter here to parse factor, the possible values that factor can take are all the values that are seen in this in site, right? So in that case, it's all 4 nucleotides plus this typo and that typo too. That's not exactly what I want to do, right? I want to enforce that the levels are only nucleotides. And so to this function parse factor, I can pass site, which is an example of one binding site, and I could pass the levels that are allowed. Now if I do this, watch, see, now what happens is that it parses it, but it warns me that there's 2 failures. One is the small g and one is the exclamation mark. And now you can see that it's replaced that little g with an NA, and it's replaced the exclamation mark with an NA. And it's basically given you a table back to say that, yeah, there's been 2 problems. A really nice thing about the tidyverse compared to base R is that it gives a lot more reporting on errors and warnings when you're doing things, and those things can really pay off. You recognize errors or weird things in your data quicker. In fact, there's a function called problems, and that if you call it with factor site, it'll tell you everything that went wrong. So you can see now, because it replaced the little g and the exclamation mark with NA, it's enforcing that it's enforcing that dictionary on the variable. Okay, so let's look at a bigger example. So honestly, this is what I typed in off the top of my head, okay? And I can put this in, and I'll get back at tibble. If I look at it, I'm going to do a nicer view here, binding sites. And it gives me back at tibble. I have a promoter, and this is the promoter, the gene that I'm looking at, the promoter just upstream of the gene. And then position in the binding site, P1 through P8. And this is the nucleotide at that position. Okay, so in the promoter of FOXA1, there's a binding site that's TAG, CC, GAT for some transcription factor that upregulates FOXA1. It's just a made-up example. Note, here I use this strange thing called a tibble, and I think this comes from Star Trek or something. It's just like this convenient way to enter data into a tibble. I mean, if there's one concept in the course that's the most not important, it would be the tibble. But basically, with this weird notation here, I put this little tilde. I can just basically enter my data in quicker, and the tibble knows that I'm intending to make a tibble that looks like this. So it's just a convenience thing. Now, it's interesting here is that I didn't intend to make any spelling mistakes, but going really fast and preparing for the class, I actually made at least 2 here, if not more. I can see them now, right? But as I was typing in, I didn't notice that I had done something wrong. So there's actually some mistakes. So, well, you know, that's a problem, right? I mean, you know, whenever we're entering something by human hands, we're going to have errors, and you want your programs to sort of point those out before, you know, you start to do a lot of analysis, right, and have all sorts of problems downstream. Okay, so what I would do here is I'm going to pipe my binding sites into a mutate function, right? So we know what mutate does, right? It creates a new column called P1 here. And well, actually P1's already existing in the tibble, right? It's that first column here. So I'm just updating that P1. I'm going to write over it by parsing P1, but I'm enforcing here the levels as nucleotides. So when I run that, when I run that, I can see here that it says there was one problem with the small t, and that makes sense, right? Because if I come back up here, I have this small t there, right? Yeah, so it caught my error, right? But that's just for P1. How would you then do this across all 8 columns of my tibble? Well, again, the beginner way to do this, right, which, you know, it's okay, but it's tiresome, is I would just copy this line 8 times. You know, I would do, you know, 8 times. I'm not going to do it here, but I would change this to 2 and 2, right? 2 and 2, and do this until I get to 8. But that's boring. Another way we attempted to do it is to say binding sites is pumped into mutate, and I can use this across function, which says take all the columns, except here I'm specifying not promoter. I don't want to do it to this column here. I just want to do it to those guys. So I can say not across all columns, except the promoter, and I call parse factor. The problem with this is that I'm not enforcing the nucleotides. It'll work, but it doesn't complain that these guys are not nucleotides, right? So how do I pass then both binding sites and the level information, this levels is equal to nucleotides, to the parse factor function? It's a bit tricky, right? So this is how you would do it, or one way to do it. Same as before, I pump into mutate across everything but the promoter column. I don't want to do it to this guy, just all these guys. And now I write this weird, so this is modeled, you read this in R as is modeled as, and so I call parse factor. So just you have to trust me, you put that there, and then you call parse factor as you did up here. That doesn't change. And now use this notation .x, and that's going to refer to binding sites, okay? And now if I put .x in there, I can put past the second parameter to parse factor, and then save the result to binding sites. So it starts off as binding sites, it changes some things, and I save it back to binding sites. And when I do that, I'll see that it gives me, in fact, my warnings that I have a T there and I have an exclamation, a percent sign. So that seems to have worked. And if I look at this, I should see that it's filled in NAs. So it's enforced that rule, and everything here is a factor, except of course promoter. if you get nervous about this, I don't blame you. I think you have to look up something in Google Chrome, like how would I find this? I would probably be something like pipe in tidyverse, two arguments, something like this, right? And then you would get maybe a link to some examples, or like how to use piping magret with multi-argument, etc. And you would just spend a little bit of time, not surprisingly, at Stack Overflow, and you'll find a solution, right? And of course, if you don't, you can always ask Stack Overflow. And I think I'm gonna spend a few minutes in next lecture talking about how you should do that, actually, but because there's an art to it. Okay. Okay, so now returning back to our Tara Oceans data, and this problem with size lower and size upper, I can easily make size lower into a factor. I can just do that, and I can see now I have my 4 levels for T1. And if I want to update my tibble T1, I can do this call where I take T1, I pipe it into a mutate, and I create a new column, new variable, but actually that variable has the same name as the old one. So all I'm going to do here is overwrite the old one with size lower as a factor, and I'm going to take that updated T1 and save it back on T1. So it's okay. So what's interesting here is that this is writing over size lower, right? And T1 is writing back over itself, right? So it's just a way of updating the table in place. I don't need to create a new tibble to do that. And when I do that now, and I come down here, I can see that size lower is now a factor. Now the problem was, second problem was that size upper is numeric. It's not a factor. And so I should make it a factor. And what I'll do is I'll first make it into a character, and then I'll make it into a factor. Okay, so now it looks just like size lower. It has, I guess, 5 different filter sizes. And the same thing, I'll update T1 again as being the result of piping T1 into mutate. This time size upper is updated with T1 size upper made into a character and made into a factor. And so when I do that, now what I'm going to see is that both size lower and size upper are factors. Okay, so that's table 1, and that's, I think we're in pretty good shape. Now there's other things in here that we might want to consider. Some of these fields are still not completely very beautiful, and the levels are not very beautiful. Let me show you an example. This pelagic biomes, these are quite long, so these are the levels within that variable called pelagic biomes. Now, if we were plotting these things in ggplot, they might be a little bit problematic, and we might want to abbreviate those. I won't do that right now because it's a lot of work, and if we're not going to use those columns, then it's not really worth it. If you were working on your thesis or a company project and you're going to use this data over and over again, then you might want to consider making that investment to really clean up the T1 tibble. Okay, so but we're going to move on for time reasons to the 6th sheet from the Sunagawa data, and that was called table 5. Again, README was sheet 1. So here I'm going to read it in because we'd saved all of those earlier, and I can look at that at my new table T5, and if I look at T5, I can see here that we have this Pangea sample ID, and the question would be, what is the relationship between T5 and T1? So I would suggest you stop the video right now and take a look. Otherwise, if you're back, then if I look at T1 Pangea, I can see that Pangea in T1, that whole column really, I can just show T1. This Pangea ID here is the same thing, it seems, at least on the surface, as T5's Pangea sample ID like this, right? Now you got to be a little bit careful here because there's also this sample label that has a slightly different structure. But if I look at Pangea, whoops, T5, what was it? T1 $ Pangea ID 1, I can see it has basically the same kind of structure as what we have here in table 5. Now that's really, really important concept because these are the keys, what's called a primary key, that's going to allow us to join table 1 and table 5 in a few minutes. Again, I don't like the titles of table 5. They're quite long, and they again use these backticks, which is a bit annoying. Now the table's not very big. T5 has only something like 1, 2, 3, 4, 5 columns, so that's pretty easy. I'm not sure what these columns are. They're measures of the ecologic diversity, actually. These guys are all measures of ecological diversity, and this guy is too. It's using this my tag that we talked about a couple lectures ago. This is the number of unique 16S regions that were detected in that sample, so it's also a measure of diversity. Table 5 seems to be different ways of capturing diversity ecologically. So that's really interesting information, and we want to keep it around. But maybe what we should, I see a typo there. What we want to do is change the titles, just like I did before, into something a little bit nicer. And when I do that, that looks a bit better. The 16S, why does it still need a backtick? I'll leave that open for you to figure out. And so there's nothing else to do with that table, I think. I think it's looking pretty good. I don't see how it looks tidy, right? I have no problem with it. These are all doubles, and we're good to go. Okay, so then table 8, which was sheet 9, the last guy, it also has some interesting information. The other tables we're not going to look at here. They have other information, but we don't need them here, and this suffices. This table 8 has a lot of chemical data, so I'm going to read that in, and I'll show it to you. Table 8 has a lot of chemical data, so let's see here. What you get, again, see, the question is, what are the keys here? And again, it uses this Pangea sample ID, same as table 5, right? That's good news. That's going to be our primary key in this table. I'll come back to discuss that in a little bit. And it's going to be the foreign table when the foreign key when we want to join these tables. We're going to hear this words over and over. But before we go on, we can see that there's data like the mean latitude and longitude, the depth, temperature, all sorts of things like in here, mean salinity, oxygen, depth, depth max O2, and then stuff I don't really know even, but you know, a lot of different data. So it could all be very interesting. I'm going to update just a few of the column names that we're going to use today. Some of them are quite long, and I'm not going to go through and do every one. Again, you know, if this was your heart and soul of your research or your company, then you would probably take the time to really do everything, right? But so now you can see that more or less, like, these names are a bit nicer. Sometimes you still have this, because of these square brackets, R is still insisting on the back tick, but that's okay. It's you know, it's not a religion. It's just you know, it's usually, it's good principle, but it's okay to have some of it, right? But mostly the columns are cleaned up, and then these guys down here I didn't touch. They're still a bit messy. Okay. All right. So now you know, we have tables T1, T5, and T8. And if I look at T1, I can see that it has this Pangea ID that we already said corresponds seemingly to table 5. It has this now Pangea ID. So it's the same, that's the same ID, the same information in both tables. And now T8, if I go there, I can see now it also has this Pangea ID. And so that's really key here of linking these three tables up. And That's called a key. Now, in fact, Pangea ID is a primary key in each of T1, T5, and T8. And that means that it uniquely identifies a row. That's an important concept. Would date be a good primary key? Well, date might be okay because it would be basically impossible to do. No, I take that completely back. Date would not be a good idea because you might do more than one sample on the same day. So if you said, okay, 2011 415 was my primary key, that would mean that there can only be one row in the table, one observation with that day. Now, if somehow you only did one sample a day, then that would be okay. But clearly we see here that in some cases, like here, that they've done more than one sample in a day. Now, would for example, time and date work? Apparently not. These two guys here, they seem to be done at two different. Oh, see, they're done at two different depths at the same time, and so they're not the same entry. They're at the same latitude and longitude, of course, because they're in a boat, and it's the same moment, but the depth that they measured at are different. So, date and time would not be a good primary key. Salinity would not be a good key either, because it could be two spots in the ocean that have the same salinity, or the same spot and two different samples taken. Okay, so this is probably designed to be a primary key. So that's going to be unique to the study, and that's probably basically some kind of random number that they chose because it's just unique. All right. Okay, so now we have to figure out how to join all of this information into one table, and there's different ways to do this in the tidyverse, and I have some notes to show you. A tidy data, everything should be in the same tibble, and as I was pointing out here, what we have is that this column corresponds to this column, which corresponds to this column across T1, T5, and T8. So we're in good shape. So now it's going to be quite straightforward to link this data, and Pangea ID is both a foreign and primary key. Primary means that it's the key, the unique identifier within a table. The foreign means that it's what connects us between them. Okay. So let's start with tables T1 and T5. And here we have a Pangea ID. It's just an arbitrary number, Tara underscore X, blah, blah, blah. And in T5, we have Tara B1, blah, blah, blah. Now, the order of these identifiers is not necessarily equal to the order of identifiers over here. We don't have any condition that these guys are ordered in the same way as here. And in fact, they may not even be the same set of samples entirely. In fact, we can check that by using set operations. And if we, do I have them down here? So what I can do is I can come over here and back into my R session and did I show these? Just do that right here. So I can do this, intersect. Well, firstly I could say the length of T1. No, I'm going to do n row of T1. That's the number of rows in T1. And n row T5. That's the table we're looking at, the second one. And I can see that already they're not equal. So there's more rows in T1 than in T5. In fact, I can ask, how do they intersect? So what's the intersect of T1 Pangea ID and T5 Pangea ID? And this is, I'm not asking for T1 and T5 directly. They don't have to be equal everywhere. I'm just, because we know that the two tables, these columns here are not equal to those columns. I'm just asking how many of the guys here are also in there. And that's where I would use intersect. And intersect is a bit like an AND gate. Okay? And I can see that there are 137, 8, 9. There's 139 identifiers in common between those two tables, right, in the intersection. And I can say also set diff of T1 Pangea ID and T5 Pangea ID. Now, set diff, right, in this case here it's T1 first and T5 second. That's basically asking for this area, that's T1 minus difference, so difference, the set diff means difference. So it's T1 minus T5. We get rid of T5. So we're going to get back this number here, the number that are unique to T1. That should be 104, right? 2, 3, 4. It's going to be 104, right? Okay. Now, that means though that we can already see if we go back and play detective here, we knew that there was 141 rows in T5 and there's 139 there, so it should be 2, right? So let's see what they are, because I mean, I think at this point you might say, well, why aren't they equal, right? Like, why is it that T1 has more samples than T5? I mean, T5 was a measures of richness, and T1 was all sorts of different factors about those samples. Why didn't they have their measures of richness for those samples, right? So if I do that, I get something really weird here. Total and mean number of repetitions. And there I'd say, uh-oh, that's really strange. And so if I come in here and I see the tail of T5, this is the bottom of the tibble, it had 2 extra rows that a human had entered that were no longer Tara IDs in table 5. They were like kind of a summary of the totals overall and the number of repetitions or something. Okay, so some human had written those things at the bottom, and the data was just not like the rest of T5, right? So you can imagine, you know, they just annotated that extra information at the bottom, and now it's kind of screwed up our analysis. So we need to get rid of those two. So what I'm going to do is I'm going to apply a T5, pipe it into a filter, so filters gets rid of rows, and I'll do a string detect on the Pangea ID for Tara. Now, of course, these 2 rows down here don't start with Tara, and so it won't get picked up by the so any row that has Tara will be kept, and any row that doesn't will be filtered, and I'll save all of this back to my tibble T5, and when I do that, and I do a tail of T5, lo and behold, I am now at it's gotten rid of those last 2 rows, and overall I have 139, not 141, and so in fact this is just a mistake, right? That's actually, in the end, that is a 0, right? So in other words, T1 is a superset of T5. So all the information, we have everything about, you know, what was it, 104 of the samples, and we only have partial information for about 139. That's okay. So in general, you might have something like this where you have a row like this, T0 with a value 5. I should have put value in the other one. Let me see here. And this might have a value of A, and this might have a value of B, like mine. Okay, and this might have a value of C. Okay, so T0 exists here, and it exists there, T1, T1, but there's no matching T2 in this table. Likewise, there's no matching T3 in T1. Okay, so that's a little bit more graphical here with colors, right? Same thing, nothing's changed so far. Now, a join brings those 2 tables together based on the key. Okay, so how does it do that? Okay, so there's different options, but this is the general concept here, is that you say, okay, well, T0 maps to T0. That's going to be easy. T1, T1, likewise. But T2 and T3 are not matched in the other table. And so those are those ones what you do with those cases is what changes. So the first idea is the inner join. And in the inner join, it's like an AND operation because it says, hey, the key must be in both tibbles, otherwise throw it away. And so that's easy, right? Because T2 is not in this table and T3 is not in that table, we just keep these two guys here. And now what happens is that we have a value from table 1 and we have a value from table 5. So T0 has the 5 from here, and T0 has the A from here, okay? So the inner join is like an AND gate, no problem, right? A second option is to say, no, I want to keep those observations that are, only in one of the two. Then there's three options. The simplest one is the full join. It's like an OR gate. It's the most liberal. Okay, so it basically says T0 matches to keep T1 for sure. T2, what we do is we create a new row in this table, T5, make it NA for the key, and make its value NA. Likewise, T3 is unmatched in this table, so make a new row, call it NA, and an empty value. Now, in a full join, you simply take everything, all four of them, 0, 1, 2, 3, they all show up. T2, which came from table 1, has a 3 here, this guy, but now it has an NA because it didn't have a value in this guy, okay? And T3 brings over an NA because it only had a value in that table. So you get NAs in your table, but all the rows are kept. So that's the outer join. Then there's mixtures, okay? A left join and a right join. And as you can imagine, they allow you to say, okay, a left join says, I keep anything in the left. All these guys will remain, okay? I'll keep everything in this table, and I'll keep everything from this table that's got a matching key, and anything that doesn't, like T3, I throw away. So here I'm going to get T0, I'm going to get T0, T1, because they match, easy cases, and T2 stays, and it gets a 3, it gets that 3, but now it has an NA there. This guy is thrown away, it's not kept. And you can imagine now the right join is just the opposite. It says, essentially, now that basically, hey, this guy here matches to an NA, but T2 is not going to be kept, right? So now I'm going to put an NA in here for this guy, T3. It'll be kept on the right join, and the C will be there, but this guy here will not be kept, right? Okay, so that's the left and right join. Okay, so let's go back now to our situation up here, and we could try all the inner and outer joins. For example, here, if I do an inner join, I get back something like 139 rows, right? So why is it 139? Again, if we go back to our Venn diagram, blah blah blah, where is it? Right there. The inner just keeps those guys like the AND gate that are in common, right? Now the outer should be 243, right? And the left should be, what would the left be? It would be 130. It would be the same as the outer, right? And what about the right join? What would the right join be? So you could maybe stop before you type those guys in and make sure that you understand, you can guess how many observations will be in each of the resulting tibbles. So it begs the question then, what join do you use? And we're going to do a full join to keep all of the information. Okay, why? Well, if you do an inner join or a left and right, then you're going to throw out some data. And if you're throwing out data, you should make sure there's a good reason for it. So here I've called the result of my full join T15 to represent the join of those two tables. if I use the inner, I'm going to throw out 104 observations here, and I don't know if I should really do that, right? Why throw out that data unless I have a good reason for it? Now, the bottom, I can foreshadow here to tell you that these samples here, these 104, don't have sequence data for them. It's just these 139, and that's why you don't have these measures of ecological diversity. So you still have measures of the salinity and depth and temperature of the water, so you have some some data points on these samples. You just don't have everything. So it depends on the question that you ask downstream whether you're going to keep these guys or not. And so I think at this point in the analysis, you just say, okay, well, I keep everything. Okay, so then we have table 8, T8. So I can do the same game here. I'm just going to copy and paste these all in, and we're just doing our detective work. So now, let me back up. Here it is. Table 8 has 245 entries. Table T15 has 243. There's 2 extra rows in T8, so I suspect that there might be the same kind of problem that we observed before for table 5. Could be coincidence, but if I ask the intersect, I see that there are 243, so there's just 2. The set diff is like, yep, there you go. So here I've asked the set difference of table 8 without table 15 on the Pangea ID. So what's in table 8 that's not in table 15? And it's 2 rows, 2 observations that look like comments in English about the values being this or that or some other note. And so we need to get rid of those guys, and that's exactly what I'll do here. It's the same thing as I do above. I just get rid of those last 2 lines by filtering on all the strings, the Pangea IDs that begin with Tara. And when I do that, my table 8 and my table 15 are going to have the same number of rows, and they're going to agree on all of their identifiers. They're both the same size because the intersect now is basically full, right? It's 243. Yeah, it's 243. So all of the identifiers are the same. And so I'll just do another full join between table 8 and table 15 to make table 158. And table 158 is now a very big table that has a lot of data, right? So now you can see that it has 243 rows. That's the number of samples. That hasn't changed. But now we have 58 columns, everything from the date, latitude, longitude, all the way down to the Shannon diversity and richness, etc. What was in table 8? I don't think, did we look at that? Table 8 has a lot of, this is the original table 8 before it was joined. It has latitude, longitude, depth, salinity, oxygen, nitrates, a lot of physical chemical parameters of the sample. So it's a super interesting table. Okay, one last note here is that in this tibble T8, I had a latitude, a variable called latitude, and if you remember in table 1, there was also a variable called latitude. So presumably they're equal between the two, but R doesn't really try to figure that out. What you'll see now in the full table T158, if I look, I see that there's a latitude of X, and there'll be further down something that's latitude of Y. Where is it? And now for the life of me, I cannot find the latitude of Y. I can find depth. Oh, there it is right there. Latitude of Y. Okay. So R said, hey, okay, this table on my left, it has a latitude column, and the table on the right during the join has a latitude column, so I'm just going to call it latitude X and latitude Y. And so you can see both values. That's all it does. Okay. So now we need to download the 16S rRNA count data from Sunagawa et al. So recall from a couple lectures ago, we found that down at the bottom of the web page, their count tables available. So basically, for every OTU, operational taxonic unit, just think of this as a taxon, a species. They give a number. They give a count for the number of 16S rRNAs they found for that species. Now, this file here, this is the 16S rRNA actual sequences. It's a pretty big file, so we're not going to download it. We don't really need it. That's the actual rRNA sequence for each of the species. I have one example of it here for you. Where are we here? Just go to the end. Yeah, here it is. So this is the, if you download this data, you'll get one file for each Tara Ocean sample. And if you open that file, you'll see that there's literally millions of these guys for each site. And this is one read from the sequencer. And this is the rRNA that was sequenced, right? This guy here. And that rRNA identified a species. And so that's what the count matrix, this count matrix here, is made from. It basically counts how many times that specific rRNA was read in the sample, and that rRNA corresponds to a sort of a signature recall of the species. If you can go back to the slides from 2 lectures ago that I made by hand explaining how 16S works, and you can certainly ask me questions in the lecture if you have any. Now, I could have downloaded, I can then again right click on this, right? And I can copy the link address. Whoops, I mistakenly started the thing. I should right click, I guess, in a PC, is copy the link address and then go back to my RStudio, and I would go to my terminal window and I would go into raw and I would do my wget with that address, and if I did that, it would download the file. I'm not going to do that because I've already done it, because it takes a few 30 seconds or so to download, so I won't do that right now. Instead, I'll just show you that in raw it's there, and the thing is that when you download it, let me just show you again the file name. It has a .gv on it, and that means it's GNU zipped. It's a compression algorithm that made that file smaller. So you need to use something called a gunzip or GNU unzip here on that file, on the compressed one, and then it will have an extension that's just basically TSV, which is tab separated values, right? Which is what we want to import into R. Now, how did I know to gunzip? I didn't. What you do is you go up here and you say something like how to unzip .gz file in Unix, and you will see right away that it says gunzip, and you're good to go. So you'll figure that out. Okay. Now, I like to put those commands that I used right in my R Markdown file because, again, it helps to make reproducible analysis, right? You can see exactly what I've done. All right. So I'm going to read that file in. It's just a read TSV command, and my file path goes to the raw path like I did before. It's up above in the document, and that's the name of the file. When I read that, whoops, I'm not in R. There, I'm in R now. If I look at that, I have a tibble, abundance, because it's the abundance of all the species, and it's big. It's 3,565 rows, observations. So each row corresponds to one taxon. So one's basically a species or a strain. Sometimes they couldn't maybe identify exactly what species it is, so they could just say what genre or class, kingdom it belongs to. But generally speaking, every species that was identified is a single row. And the first row here is just like unidentified. So basically they got a read back from the sequencer and they didn't know what species it came from. That's sort of microbial dark matter in a way. That's what we call it. My physicist grad student loves that name because he's, physics has this dark matter, right? Okay, so yeah, and the columns here are the Tara Ocean samples. Okay, so there's going to be 146 of them in total. No, 104. Well, there's 146 columns total, but 7 of them are used to describe the species. Everything from the domain, like bacteria, and then like the phylum, so proteobacteria or cyanobacteria, etc. And then, inside of proteobacteria, you have like alpha bacteria and beta bacteria, beta, gamma, delta, epsilon bacteria, etc. And then, and then it breaks down and breaks down and breaks down. I can show a little bit here, like if I say here abundance at class. Well, it'll be very long, but I can do something like unique abundance $ class, and I typed that wrong. Abundance. I can see that there's 148 different classes of species that were picked up. Methanobacteria, I don't know everything. What's it? The chlorobia, I don't know. Acidobacteria, which are usually, important. Nitrospira, they're very big, frequent in these data sets, etc. Okay, so the main thing, right, is that you might want to pause the video and make sure that you understand how these rows together, each row defines one taxa. I don't want to use the word species because it could be a strain or a genre, but one taxon, and then this is the number of times that taxon was detected in this Tara Oceans sample. So, for example, what that says is basically 5,000 reads were not identifiable in Tara 018. Okay, but 1,021 belonged to this bacteria, presumably that's proteobacteria, that's proto-alpha bacteria, that's probably Robacter, and then I'm not sure what else. So, okay, good. Hopefully everybody's understanding that. Where were we here? So now we need to ideally join the count matrix with our T158, right? Our master metadata. But now the question is, how do we do that, right? Because, well, what is the what are the keys, right? How do we match this table to T158? I'll just show T158 again quickly. So what matches it? And you recall that when we looked at T1, there was a second ID, this sample label here in T1. And we had a Pangea ID down here. And we used this Pangea ID to join the previous tables. And now our focus is going to be on the sample label. Now, that's one problem. So the sample label here, I think, matches the column names of our abundance, right? These guys. But, that's a problem because these are columns, not rows. In T158 and T1 and T5, T8, these were rows, observations. And now they're variables. So how do we get this abundance tibble into the proper format? So you might want to pause and just think about that a little bit and see if you can solve it. Well, you would probably use pivot longer. And this is the basic idea, is then to get these guys down, to make this table longer by turning these guys into rows, right? Into observations. So here with pivot longer, I say, give me all the columns that start with Tara. That's all these guys, right? Not these guys, but all of these guys. And I'm going to create one column called sample label. That's going to contain all of these different guys. And the value is going to be called count. maybe it's just easier to show you this. When I run this, it takes a while, but that was not too bad. Now what you'll see here is now I've only got 9 columns. I went from several hundred down to just 9. How many rows do I have above? This is just the tail. So if I look at abundance in total, I have a huge number, almost 5 million rows. Okay, so this undefined, 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 there's a lot of them now. There's one for each of the 143 Tara Ocean samples, right? So I've really gone from a really wide matrix into a very long one. But now what I want to do is I want to effectively get these guys as my variables, right? And I want to keep these as my rows, but I want to put these guys as my variables. So I'm effectively like transposing the matrix. Okay, so how do I do that? Now I use wider. So now what I'm going to do is I'm going to say my names from are all of these guys from domain up to OTU rep. And what it's going to do is glue those guys together into one name. And it's going to put all those values, get its values from count, and set that back equal to abundance. So let me show you when I run that. Okay, so that's going to take a few seconds. So what we hope to have here is that all of these guys now become a single column. And it's going to contain the count in that sample for that guy. So let's see what it looks like. Now, I have a lot of columns. Now, I'm back to having 139 rows. That's my samples. That's just one for each Tara Ocean sample. But now I have one column for every species that was identified, so undefined, etc. And so now I've essentially transposed the rows for the columns and the columns for the rows. Okay, so now I have a chance to join these two tables. Now, one thing here is that I should say something about these column names, because they're a little bit weird. Now, what R has done is concatenated all those 6 fields. So now we see that it's bacteria. Okay, it's undefined, 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 unclassified. That's boring. That's the first guy. But the next guy is bacteria, proteobacteria, alpha proteobacteria, Rhodospiralis, and then a species within the Rhodospiralis, and then some that's a kind of a technical thing for the strain or something like that within that species. And all the column names kind of look ugly like that. Now we could clean them up by doing that separate function, but I'm not going to worry about that for now, because that information is there. It's not necessarily, it's going to cause problems if we start to plot, but at least we know that sort of phylogenetic trace for each species. Okay, so now we're in a position to finally join T158 and the abundance, our new abundance matrix, right? And I can do this right now. I'll do the usual little detective work. In T158, I have 243 rows, 139 in abundance. See, that's, again, I think those 139 have been sequenced, and the other, whatever that is, 90 or something, 100 of them, 104, they haven't been sequenced, but we have other information for them. And if I look at the length of the intersection between the two, I see that there's 139 in total. So now, basically, if I look at my abundance matrix now, I see that it has this sample label variable, and I'll use that as my key to join. And when I do that, I'm going to get this really beautiful table that contains all of my information. Done. Thank God. So let's take a quick look at it. It's a massive table, but this is a massive international project. So you have now 243 samples from the Tara Oceans project across roughly 35,000 species that were detected, and then about 100 different variables. All of these guys up to Shannon diversity. They're oops, sorry, I didn't mean to do that. All of these variables up to here are basically metadata, physical chemical properties, and then all of these guys below are the different species of bacteria that were detected in those samples. Now the final step here is to save these files, to save the data structure to file. There's two ways to do that. One is to save using the comma separated value file, and if you were to send this to your collaborators, like slow throughput biologists, then you'd use a CSV file, and that's just going to make it kind of convenient for those guys who don't know R to load it back into Excel and screw up your life. The other way, the more fancy way to do it, and what you want to do, is to save it as a RDS file. The RDS file saves it as an R data structure that it's a bit safer, because sometimes when you save to a CSV file, you lose some information when you bring it back in, and the RDS file, it's just an R data structure, so it's the best thing you can do. Okay, so that's now we have essentially all of the Tara Oceans data into our one T_all tibble, and that really is the end of the import process and tidying process.